Welcome to the Carbohydrate Metabolism Part 2, focusing on the digestion and absorption of carbohydrates. These are the learning objectives that will be covered during this session. It comes in two parts. First, working on the overall stages of digestion, followed by specifically how the carbohydrates are absorbed. The proteins and fats and minerals will be done in a separate session. This session will only focus on carbohydrates. So let us first start with the stages of digestion. So overall, there's about 7 liters of substances secreted daily throughout the GI system. This picture shows the different stages, starting with the oral cavity, going down to the swallowing phase. The oral cavity, the cephalic phase, is where the chewing is occurring with the saliva, followed by the swallowing into the esophagus, and then down into the stomach. The stomach is where majority of the digestion is going to occur, and then small intestine is where the majority of the absorption of the nutrients will occur. So in the saliva, it's relatively low sodium and chloride. There is a higher potassium and bicarbonate concentration here, and there are numerous enzymes here as listed in this slide that will help with the digestion process. So starting next with the stomach, this secretes about 2,500 cc's a day. There's a lot of hydrogen ions from from the hydrogen potassium ATPase proton pump. The parietal cells also secrete chloride and bicarb at the mucosal surface to protect it from the acidic environment. And then the intrinsic factor is also secreted from the parietal cells. The chief cells are doing most of the digestive enzyme, which is pepsinogen, which gets broken down into pepsin. In the pancreas, here there's also a high amount of bicarb that is secreted to increase the pH. And then the major enzymes here, which will be covered in just a little bit. Moving on to the small intestine. Here it's or the small and large intestine, but mainly small. There's about 1 liter of fluid that's secreted here. And here also high in bicarb and other enzymes known as enterokinases or enteropepsidases. This is also where there are many brush border enzymes to help digest the sucrase, the lactase, and other carbohydrate cleaving brush border enzymes. GIP is also involved here. This is known as glucose-dependent insulinotropic peptide. This will be covered in more detail when we talk about diabetes and insulin and the actions of insulin. But GIP acts on the beta cells in the pancreas to increase insulin in response to dietary glucose. Okay, now how is this all regulated? So in the saliva, it's mainly regulated by parasympathetic and sympathetic forces as described here. There are numerous parasympathetic arms that are activated, including acetylcholine and VIP, and then further down, which have effects on water and ion secretion. The sympathetic system is mainly going to be the opposite of the parasympathetic, so it will inhibit saliva production, and it will also decrease saliva volume and flow. Once you get to the stomach, where there are vagal neurons and gastrin that are involved here, there's a numerous series of steps that occur here. Acetylcholine is going to stimulate the muscarinic receptors in the parietal cells. This will increase acid secretion. They also synapse on the enteric system in the nervous system to stimulate the ECL cells to secrete histamine and also GRP. So this can also inhibit somatostatin release. Somatostatin is an inhibitor of all of these other hormones. So this is sort of an autoregulation that's occurring here. Gastrin is increased when you eat and with vagal stimulation. And this also stimulates the parietal cells to release hydrochloric acid, and it can stimulate the ECL cells to release histamine. So this table just summarizes the past few slides, summarizing it in a way as an overview, with further details in other sessions. Now here is a summary of some of the secretions that are happening, some of the enzymes along the way during digestion. So you can see the oral cavity here with amylase, heptachlorine, lysozyme, lactoferrin, and peroxidase, and the different functions of all of these things. In the stomach, bicarb is the buffer. Hydrochloric acid is what is breaking down the food into smaller molecules. Pepsinogen gets converted to pepsin, which is what breaks down proteins. And then intrinsic factor is being released by the parietal cells to protect the B12. This binding has to occur to B12 in order to allow it to get absorbed further down in the ileum. So this picture here shows all the regulatory and inhibitory and also stimulatory mechanisms at the different areas extending from the stomach all the way down to the intestine. Further down in the intestine, there's going to be bile secreted, and it's mainly for fat absorption further down. But for carbohydrates, the big one here is alpha amylase, and this is designed to break down starches. This will be covered in just a further down. Also, endopeptidases, who are also involved in breaking down these peptides from the stomach. Lipase, as mentioned previously, also helps with fat absorption and digestion. And then we also have here the enteropeptidases that are in the brush border in the duodenum, which converts the trypsinogen to trypsin and buffers the hydrogen peroxide made by human cells and bacteria. Now let's talk about carbohydrate absorption. This is the major part of this session after covering digestion. So the salivary glands secrete salivary amylase, which breaks the starches, as shown here in the upper picture, into maltose. The polysaccharides get broken down mainly into the maltose molecules, which are disaccharides. In the stomach, the salivary amylase now becomes inactivated by the acid. The pancreas then further releases amylase as the food moves down from the stomach, and this now will break the starches also into maltose. The small intestine then has brush border enzymes, which can then break down these disaccharides into sucrose, lactose, and maltose. The brush border will break down the disaccharides, which are sucrose, lactose, and maltose, into the monosaccharides, which are the fructose, glucose, and galactose. So now the second picture is showing one of the intestinal villi cells. So on the apical side, the SGLT1 will be a secondary active transporter that can transport glucose and galactose with two sodium cations into the cytoplasm. In the cytoplasm, this galactose and glucose is then released from the 3-sodium, and they are now, the 3-sodium are now exchanged for 2-potassium via sodium-potassium ATPase with the energy from ATP. GLUT5 on the apical side is what transports fructose into the intestinal cells. This fructose, galactose, and glucose now come together and are transported by GLUT2 on the basolateral side, which is the glucose uptake transporter 2. And this is done by facilitated diffusion, so it's based on concentration gradient at both of these transporters. So this concludes this session on carbohydrate digestion and absorption with an overall overview of also the digestive process. Please subscribe to the ACM channel, for the YouTube channel, for further updates on new content. Thank you.